This is the fifth video in a series about the Golden Gate cloning DNA assembly method. In previous videos, we learned how type 2S restriction enzymes allow one-pot, one-step assembly of Golden Gate parts. We also learned about the Golden Gate Moclo assembly standard and how to make new standard parts. In this video, we're going to show how to assemble a complete transcriptional unit into a level 1 acceptor in a single step. The Moclo assembly standard, defined in detail in the third video in this series, defines a number of specific standard parts that correspond to specific parts of genetic grammar, such as promoter, 5' untranslated region, coding sequence, N-terminal tags and signal peptides, C-terminal tags, 3' untranslated sequences, and terminators. Adjacent sequences can also be cloned into a fused part, such as promoters and 5' untranslated regions, or 3' untranslated regions and terminators. As we have learned in previous videos, the overhangs created by the release of these standard parts from their level 0 backbones with the type 2S restriction enzyme BSA1 allow one-step assembly. For example, a promoter and 5' untranslated region module will fuse to a coding sequence module via AATG, which includes the ATG start codon, and a coding sequence module will fuse to a 3' untranslated sequence and terminator module via GCTT. If we take a close look at the plasmids involved, we can see how that works. The level 0 plasmid containing the Pro plus 5U standard part is in green. The level 0 plasmid containing the CDS part is shown in blue, and the level 0 plasmid containing the 3U plus terminator part is shown in brown. As per the Moclo assembly standard, each part is currently cloned in a spectinomycin-resistant plasmid backbone. We are going to show how they can be assembled in a single step, in the correct order, in a level 1 acceptor plasmid shown in red. Within their level 0 backbones, all the standard parts are flanked by pairs of BSA1 recognition sites, GGT, CTC, in opposing directions. The level 1 acceptor also contains a pair of BSA1 recognition sites in opposing directions, flanking a LacZ cassette. In the one-step digestion ligation reaction, the BSA1 enzyme will release the parts from their backbones, and also release the LacZ cassette from the level 1 acceptor backbone. The compatible ends will be stuck together by the T4 ligase to make a complete transcriptional unit. In the level 1 backbone, the complete transcriptional unit is now flanked by BPI1 recognition sites. However, instead of releasing with the same overhangs, digestion with this enzyme will release the complete transcriptional unit with a new set of overhangs, ready for assembly with other transcriptional units into a multi-gene construct. In the last video in this series, we'll learn how the overhangs in level 1 constructs allow multi-gene assembly.